Welcome everybody to an OpenEye webinar. Good morning, good afternoon, good evening. Today we'll be joined by Paul Hawkins. Paul is a product evangelist for OpenEye, and he's going to talk about shape and color, a unifying principle for modeling molecules. During the talk, if you've got any questions, please use the Q&A section in the top right corner of your screen. We'll get to the questions towards the end, and if we don't get to them live, we will get to them by email and answer them after the talk. Without further ado, I hand it over to Paul.
Thank you, Gunther. Welcome, everyone. As you said, my name's Paul. Today, shape and color as a unifying principle for modeling molecules relevant to drug discovery. So modern drug discovery could be thought of as a three-stage process. We're going to find a protein that's involved in human disease, find pockets on that protein that we can bind some ligand to. In the movie here, a small molecule ligand, and then design ligands that are going to bind to that pocket on the protein, modulate the activity of the protein, and give a positive readout on human disease. So there, drug discovery in less than 30 seconds. You're welcome. So when we were quite some time ago thinking at OpenEye about how we would want to model molecules relevant to this problem, we decided on really that there are only two we need to think about, two fundamental molecular physical properties. And those are molecular shape and molecular electrostatic potential. And we focus on these two primarily is because we believe there is complementarity in these things, shape and electrostatics in the protein and shape and electrostatics in the ligand, that drives the primary event we're trying to model, which is protein-ligand binding. So all we have to do is model molecular shape and molecular electrostatics. So starting possibly with the easy one, modeling molecular shape has long been thought of as modeling a series of hard atom-centered spheres, which works beautifully if your molecule is diatomic, as shown here. But unfortunately, most molecules in drug discovery are not diatomic. And as Michael Connolly showed in his seminal work from 1985, 40 years ago now, modeling molecular surfaces and molecular volume as a series of intersecting hard spheres becomes incredibly difficult mathematically. The calculations become very slow, which means the atom-centered sphere model doesn't scale well, either with the size of the molecule or the number of molecules that you wish to calculate on. So a foundational assumption we made at the founding of OpenEye is to abandon the traditional hard-centered sphere model and move to something called a spherical Gaussian. Here on the diagram, I show a section to some spherical Gaussian functions and a sphere, the sphere in green. The blue Gaussian, the soft Gaussian, and the green sphere have the same volume, but interestingly and somewhat unphysically, some of the volume contained within the soft Gaussian, the blue, lies outside the surface of the sphere. And this has all sorts of fascinating benefits for molecular alignment and molecular similarity calculation. So we've abandoned the traditional hard atom-centered sphere model for an atom-centered spherical Gaussian, and using some clever mathematical tricks, we're able to align and compute similarities between molecules at very high speed using this spherical Gaussian model. So that seems to be the shape problem solved. We represent the volumeness of an atom as a spherical Gaussian. Electrostatics seems harder. We've got electrons to deal with, so we have things like the time-dependent Schrödinger equation, some alphabet soup of quantum mechanical methods giving us some sort of electrostatic potential. We use the rest method or others to fit that potential to partial charges. Just like spherical Gaussians, partial charges are not physical, but they're very useful. We can take those partial charges and we can put them into things like the Poisson-Boltzmann equation to compute electrostatic potential. The PV equation is difficult to deal with for alignments. We can't use analytical approaches. We have to solve the equation numerically, which means it's slow and doesn't scale well. So we've decided to abandon for large-scale work the physical rigor of electrostatics and replace it with a much more familiar atom-centered approach where we put chemical properties like donors, acceptors, rings, cations, anions, and so on atoms or small sets of atoms. So we go from representing electrostatic potential around a molecule to properties on the molecule. So how are we going to apply these properties on the molecule? Well, we can actually use a little bit of 2D cleverness, some cheminformatics, specifically the rich and flexible language of smarts, which here we can use to express the acceptedness of the carbonyl oxygen of the acceptor in an ester group. Smarts can be written very generally, very specifically, so we can apply these to a wide range of different functionality. We can then represent, as I think you've already guessed, the color propensity of an atom as another atom or feature-centered spherical Gaussian, as shown here. So we can represent the propensity of an atom to be a donor, or a propensity of a group of atoms to be hydrophobic or in a ring, as a spherical Gaussian. So now we have spherical Gaussians for shape, spherical Gaussians for color. The calculations are reduced in speed a little by including, I should say, both shape and color, down to around 2.5, 3,000 overlays a CPU second. So this brings us to this unifying picture of a molecule relevant to drug discovery as a set of color features embedded in a shape. And this lets us use one of the more powerful principles in modern drug discovery, which is, of course, similarity. Just as similarity in 2 dimensions from the graph can work very well, similarity in 3 dimensions, expressed as a similarity both in shape and in color, as I hope to persuade you, turns out to have a great many uses. So we have a Tanimoto coefficient for shape, a Tanimoto coefficient for color, which we simply add together, unweighted, to give what we call the Tanimoto combo, or TC. And I will show you several applications of the Tanimoto combo coefficient for similarity across the rest of the talk. The first example of showing this I want to talk about is some very interesting work from what I'm going to call the 2 Steves, Steve Swan and Steve Muchmore, when they were at Abbott, now AbbVie. The relationship on the graph on the left is between Tanimoto combo, that's between a molecule with known biological activity and a new hypothesized or designed molecule. That's X. On Y is the probability that that new molecule shares biological activity with the known molecule, where share is defined as plus minus a log unit in affinity. And we have this rather beautiful and I think intuitively satisfying sigmoidal relationship showing that as Tanimoto combo between the query molecule with activity and the new molecule rises in a lead optimization context, higher and higher probability that the new molecule shares biology with the existing molecule. So this leads us to a larger principle, which I'm going to show several times in the rest of the talk, that similarity in shape and color is indicative of similarity in biology in a number of different contexts. So similarity in shape and color for small molecules is the foundation of, I think, our best known tool, which is ROCS, where we represent a molecule, usually one with interesting biology, as, again, a set of color features embedded in a shape. We represent conformations of a database of molecules to be screened in the same way, a set of color features embedded in shape. We rigidly align and optimize shape and color matching between the 2 molecules, and we score each database molecule against the query with this sum of the 2 Tanimoto coefficients, one for shape, one for color, to give us the Tanimoto combo. And again, I'm going to come back to this score several times in the rest of the talk. Now, this works, as I hope to show you later, beautifully for small molecules, but let's not forget, of course, proteins are small molecules too. So we can represent the shape and the color features of a protein binding site in exactly the same way as we do for a small molecule. And we can compare protein binding sites based on matching shape and matching chemical features. And I will come up with an example for that in terms of polypharmacology and side target effects in the next section of the talk. Now, it's been my experience that as soon as you show a drug designer, a computational chemist, protein binding site, but the first thing they want to do is to put molecules into it, to find out which ligands actually fit into that binding site. So they want to go and do docking, which is, as I mentioned at the beginning, protein complementarity, which we believe is driven by matching in shape and electrostatics, shape and color. So we should be able to use the spherical Gaussian approach, not only for protein or small molecule similarity, but protein complementarity, assessing the overlap between complementing features, one on, say, the ligand, perhaps a donor, and then one on the protein, in this case, an acceptor. And we can represent these features as spherical Gaussians. The overlap between 2 spherical Gaussians is just another spherical Gaussian. So it gives us a very quick and easy way to score protein ligand matching conformations. Given that we're using the color model here, we have limited features in the molecule, both protein and ligand, which means we have limited parameters in the model, which requires limited parameterization for the scoring function. And that means we believe that makes the scoring function robust to being applied to proteins outside its original training set. So now we can apply this idea in a couple of different ways in 2 of our high-speed docking engines, FRED and Hybrid. FRED is a pretty traditional, fully protein-centric docking engine. We have 3 stages, all of which we use scoring of poses against the protein using that overlap of spherical Gaussians. The hybrid approach replaces the middle protein-centric approach from Fred with good old ligand similarity using the Tanimoto combo between the molecule we're posing and the ligand in the binding site, usually a co-crystallized ligand. And then we take the high-scoring poses from that, pass them into the final protein-centric step. So a hybrid interleaves a ligand-centric step between two protein-centric steps. So that has two important consequences for performance in docking. One, the post-prediction performance rises significantly. On the left, fairly trivial case, self-docking. You take the molecule out, you dock it right back into its own shape and color. It works really well. Very high success rate. On the right, the rather more relevant cross-docking problem, where recent work from Bayer on a number of different proteins, all of pharmaceutical relevance. On the left panel, outlined in gold, results from hybrid. On the right, outlined in cyan, results from hybrid. Hybrid performing equivalent to or superior than Fred in every single case. So we get better results from this somewhat ligand-influenced approach. And though this part seems like cheating, we actually get the better results faster. Fred around 3 seconds a molecule, a little bit more. Hybrid less than 2 seconds a molecule. So about a 40% increase in speed and improved docking post-prediction. So great answer. So what I think I've sort of established is we've got a variety of different ways to apply the shape and color technology to a variety of problems. So I'm going to start down the journey of drug discovery with target ID and target validation, finding protein binding targets, pockets that we'd like to work against. So as I mentioned, we have the ability to represent protein pockets, shape and color. That's the foundation of our tool called SiteHopper. And that rigidly overlays and scores protein binding sites, as you can see in the cartoon. As you can also see in the cartoon, protein binding sites we fully expect to be different in volume, in shape, and in distribution of color features. So we rather than use the unweighted sum of shape and color as we do to generate the standard small molecule Tanimoto combo, we use a weighted sum here where we weight the color score times 3 to give us what we call the patched score that ranges 0 to 4 versus the Tanimoto combo unweighted, which ranges 0 to 2. So I'm going to give you a couple of examples of using this. One of my favorite molecules in oncology, that's Gleevec or imatinib. It's designed originally to be a treatment for breast cancer by targeting the BCR ABL1 kinase, a co-crystal structure shown here. Now, the original design goal for imatinib, for Gleevec, was to be exquisitely selective for ABL1. It turns out that Gleevec is anything other than exquisitely selective. It binds a lot of different kinases, and that's part of its magic, as it works very well for a lot of other cancers. So here I'm showing you results from a SiteHopper calculation where I take the ABL1 Gleevec co-crystal structure and I run it with SiteHopper against about 120,000 pockets from the PDB, looking for pockets with similar shapes and color feature distributions. The second ranked out of 120,000 non-ABL1 sequences is this protein here, KIT, the KIT kinase, which is implicated in GI cancers. The co-crystallized ligand here for this KIT structure, 6G2J, is this molecule. Not very similar to Gleevec, but it induces a binding site that's actually very similar to the binding site that Gleevec induces in ABL1. It was subsequently discovered that Gleevec is a potent inhibitor for KIT, more potent even than it is against ABL1. And a few years after Gleevec was approved for treatment of breast cancer, its original target, it was also approved to treat GI cancers by inhibiting the KIT kinase. So subsequently, Gleevec was co-crystallized with KIT, and there are the two structures of the KIT and ABL1 kinases, both co-crystallized with Gleevec. And as you can see, they line up very nicely. The molecules here match very well. And there's also a pretty high patch score, again, looking at the similarity in shape and color between these two binding sites. So that's a little anecdote of SiteHopper at work. I'm going to take a slightly larger view here. So work published from Vertex a few years ago, looking at SiteHopper's ability to discriminate between known matching binding site pairs and known negative pairs. The overall performance of SiteHopper here is excellent. Co-A in the middle there, VAUC recovery here is about close to 0.9, about 90%, so it works very well. The same data as in the graph labeled A is on the left. The x-axis is the sequence similarity between the query pocket and the known positive pocket. As we see, as the sequence declines in similarity, the sequence-dependent methods become much, much less effective, but the performance of SiteHopper is much less affected. So we see the red line there substantially above the others when we get to a sequence similarity of around 35 to 40%. So at low sequence similarity as we'd expect, the 3D method significantly outperforms traditional sort of, if you like, graph methods based simply on sequence comparison. So now I think we have a sort of an armamentarium of different approaches to different sets of problems. We can describe both protein binding pockets and small molecules as shape and color and compute similarity between them, like copper and rocks. And we can look at complementarity between small molecules and proteins in our docking engines, Fred and hybrid. So what I'm going to do now is take these approaches into the next sort of stage of drug discovery, where we're going to be looking for a protein ligand structure. The job of the structural biologist in most industrial drug discovery is to fit ligands into protein structures, into electron density usually. So the structural biologist's job is to tell the project team how a particular new molecule fits into a binding pocket, which interactions it makes with the protein to help the team design new and better ligands. So their job, day to day, is to fit molecules into here electron density from crystallography. This can be pretty easy when the density is high quality. It can be, as it often is in drug discovery, less high quality, much more amorphous, but the job is the same, to fit the molecule into density. And we can even do that, as I'll show you in a minute, for covalently bound ligands too. So how are we going to approach this? Well, we came up with the sort of realization that just like a molecule is a shape, electron density is a shape. And we know something about treating molecules as shapes. So perhaps we could fit shapes together, shape of a molecule fitting into the shape of electron density. And that led us to a method we came up with called adapted, I should say, called adiabatic optimization. So our goal here is to fit a molecule, a ligand, into electron density in such a way that the conformation, the pose of the molecule has low molecular mechanics strain. And to do that, we use this control parameter lambda, where we slowly and steadily increase the influence of shape in the final potential so that we get a good fit to density while returning a low strain conformation for the small molecule that we're fitting. And that is the basis of our engine AFIT, which works very nicely to fit into all sorts of density, example shown here, including this one. Now, this is a structure of nirmatrelvir, which is the active component of Pfizer's Paxlovid medication, bound into the active site of its target, the SARS-CoV-2 main protease. So they published this in their science paper a few years ago. We were very proud of this. This is an example of our tools at work in real drug discovery having an impact on human health. So this was really great for me to see. Okay, so now we have this magical protein ligand complex. We can get this either from crystallography or also from cryo-EM, which opens up a whole new range of large and flexible proteins liganded with small molecules. And we can take these protein ligand structures and we can consume the information in them in several different ways. We can consume just the binding site representation with CiteHopper. We can consume just ligand information with rocks, or we can consume both protein and ligand information together in our docking engine hybrid. And I'm going to look at rocks and hybrid now in the context of the next stage of drug discovery, computational database screening, finding new molecules that are going to have that biology that we want. So firstly, I'm going to look at just consuming ligand information from the protein ligand complex with rocks. So the example here from Simbi Yan and colleagues at Merck looking for new HIV integrase ligands. They knew about the quinolone series. There is its crystal conformation as a shaken set of color features. They screened their entire Merck corporate collection, several million molecules with rocks, to look for new leads with different structure but interesting biology. And they came up with this rather fascinating tricyclic core, a little bit of SAR transfer from one series to another, and some clever chemistry. And they came up with this high nanomolar binder for HIV integrase that actually went all the way to IND. So a very fast start, a very interesting new avenue for the project, produced by rock screening across their corporate collection. Now, Merck has a large corporate collection. The company itself is old. It's acquired a lot of compounds. It's merged with a lot of companies. It has a very large corporate collection. But newer companies, less well-resourced companies don't. How are they going to be able to compete with companies who are able to screen large collections of their own molecules? I think this desire to be able to have access to large and interesting chemical spaces has been one of the drivers for the explosion in the availability of molecules, vendor libraries, from companies like Enamine and Wuxi, Otava, and so on, who are producing 100 million, some cases multi-billion spaces for anyone who wants to go and search them for search. And when you add all the available spaces together, be they enumerated or unenumerated, we're getting into available spaces of in the trillions. So it's a great time to be in the virtual screening area. You have access to more molecules than you ever had before, and you can get them in a very simple way. You simply identify the molecules you want, send some money to the vendor, and a few weeks later, a box of molecules drops to your mailbox. Success. So this molecules for money model works very well. There is also, I think, a bit of intellectual flexing going on here. I screened 1 billion molecules this week. What have you done? And these large libraries actually tend to produce better quality hits. And that's nicely exemplified here for some work from AstraZeneca, from AZ, using our 3D shape and color similarity method, scoring molecules from databases of different sizes with Tanimoto combo. So on the x-axis, the size of the library screened, 100,000 to 10 billion. On the x-axis, so the y-axis, the Tanimoto combo for the top 500 molecules across about 8 or 10 different targets. The horizontal blue line is the mean across all the different targets for the top 500 scoring molecules. And as you can easily see, the score of the top 500 scoring molecules monotonically increases with each increase in library size. We're getting higher scoring hits from the larger libraries. And as you recall, the work of the two Steves tells us that we're able to get more molecules with interesting biology as the Tanimoto combo rises, as the similarity known and unknown rises. So this is great news. These larger libraries are giving us better hits. To screen these very large multi-billion libraries, we have a GPU port of rocks, which we call fast rocks, and that's on our cloud platform Orion. And we can screen 1 billion molecules on Orion in around half an hour for less than $20. And this calculation shows low variance. The time and cost is not much affected by the size, the volume of the molecule, or its density of features. So it's very cost effective. It's also very dollar efficient. Here we're looking at the in-flight compute costs on both CPUs on the left, GPUs on the right. The GPUs in flight, we're able to screen about 300 million molecules a dollar, which is well over an order of magnitude more efficient than using CPU-based hardware. So we can get answers to search in these large libraries very quickly. Is wrong answers fast or helpful? Probably not. So I just took Gleevec, again, one of my favorite molecules, ran it with fast rocks against the enamine real collection, about 97 million molecules. And here are 5 of the top 6 round hits from enamine. And as you can see simply by inspection, they're all different to one another, and they're all different to Gleevec. So we're getting a large amount of diversity in the hits. We're able to explore new regions of chemical space that we couldn't find if we were simply doing things like 2D searching. So just to remind you, we have also this shape and color-based docking engine, hybrid. We'd like to use that at large scale also. So we have similar experiment to what I showed you earlier on the right here, looking at how hybrid scores are affected by the increase in library size. Here we go from 10,000 to about 90 million, and you see the same qualitative data that we saw from the AZ approach with fast rocks. Monotonic increases in the scores of the top 500 molecules. So we're getting better docking hits out of docking larger libraries. We're getting better ligand-based hits out of ligand-based screening larger libraries. So again, looking at screening large libraries on Orion, we have the GigaDoc engine, which does both Fred and hybrid. These calculations are a teeny bit more expensive and a little bit more time consuming than the fast rocks calculations. They're going to take over 12 hours and cost something more than $10,000. And unfortunately, there's fairly high variance here. As I mentioned in the site hopper section, binding sites end up being very different sizes, and the volume of the binding site is the key determinant to the time taken for a GigaDoc calculation. So we see variability in time and cost based on the volume of the binding site. So like many people these days, when confronted with a problem with time and sampling, we decided to sprinkle a little bit of machine learning onto the GigaDoc warp approach. The GigaDoc approach, and we came up with something we call GigaDoc warp. So instead of brute force docking the entire database, we take a small percentage, say 1%, we dock that. We then build a local machine learning model to predict the docking score. We predict scores for all the molecules in the database, and then we take, say, the top few percent, 4 or 5%, and actually dock those. And that then is what we return to the user. You may have noticed I haven't said spherical Gaussian function for about 5 minutes. Well, obviously, spherical Gaussians used to generate the actual docking scores. We also have the magical Tanimoto combo is part of the featurization vector we feed into building the machine learning model. So, how well does it work? We're pleased to say that it works really rather well. In the table here, Gigadoc is the brute force docking, Gigadoc Warp is the machine learning augmented approach. You can see the costs here are around 10% of the cost of the brute force calculation, while on the right-hand column, we're looking at the overlap in the hit lists, and generally speaking, we're getting more than 90% overlap, 90% identity between the machine learning augmented Gigadoc Warp and the brute force Gigadoc itself. So around 90% of the hits for around 10% of the cost. Works really rather well. So now we have some biologically interesting hits. Now it's time to do a little bit of lead optimization. This is the difficult part. Now, as I've mentioned, we have a couple of high-speed, large-scale docking engines, Fred and Hybrid. We believe that docking for virtual screening is a different problem to docking post-prediction for lead optimization. So we have a different solution for lead optimization post-prediction, which we call ShapeFit. And ShapeFit is a flexible fitting engine where we flexibly fit a new molecule into the shape and color of, in this case, a crystallized ligand. And we use the protein binding site not to score poses, but rather to simply make sure that when we're flexibly fitting, we don't clash the new pose with atoms of the protein. So this, at the end, we end up with the new molecule to be posed fitted into, on top of the crystallographic ligand. So we have the same overlay. And as I said, we don't score the new molecule's pose against the protein. We score it with the Tanimoto combo against the ligand. And again, this gives us some rather magical looking information. In the plot on the left, we've got the Tanimoto combo between the molecule that we're posing and the molecule in the binding site, the crystallographic ligand. And on the y-axis, we have the pose prediction accuracy, the frequency with which that pose, based on retrospective data, is within a 2-angstrom RMSD of the correct answer. And that's based on over 90,000 separate cross-docking pairs done retrospectively. So this is a probability assignment curve that allows us to relate Tanimoto combo between pose and ligand and the probability that that new molecule, that new pose, is correct. This shape of the graph and the x-axis being Tanimoto combo may remind you of the work from the two Steves, where Tanimoto combo there related biological activity to TC, not here pose prediction. So with this probability assignment curve in hand, we can do something really rather powerful. We can take the Tanimoto combo, the raw score, directly out of ShapeFit, and we can translate it very easily into a pose prediction accuracy. So here, if our Tanimoto combo between a new molecule and the crystallographic 1.4, we get a pose prediction accuracy of 85%. It's 85% likely that that posed molecule is within 2 angstrom of its correct pose. So now we've moved away from the language of scoring. My docking score is minus 14.6. My Tanimoto combo is 1.4. Scores that are hard to interpret, particularly if you're not expert, into the language of probability. 85% likely that this pose is correct. Much more actionable language for chemistry to do. Now, of course, if we can flexibly fit two molecules together or one molecule to another in a protein, we can do it without the protein. That's the job of FlexiRox. An example here, rapamycin. One of my favorite molecules. It's a molecular glue, binds two proteins together, and the one binding part is for the western part, as you see there. And when we do conformation sampling and overlay with rocks, the western portion, the FKBP12 part, matches really very nicely. The effector region up in the northeast, on the other hand, the top right, doesn't match so well. However, when we fit flexibly, we of course end up with a much better answer. Looks very well. We can look at accuracy for fitting across a set of macrocycles, work published from the Jens Carlson lab a few years ago. Flexible fitting accuracy on the left here, Tanimoto combo, and on the right, RMSD. So on the left, higher is better, on the right, lower is better. And to no one's surprise, flexible fitting works better, whether we control strain in the fitting using good old RFF94 in the middle, or the rather newer and more shiny SAGE force field from the Open Force Field Consortium. So flexible fitting works better. Okay, so it's all come down to this. We've got ways of aligning molecules to one another flexibly or rigidly in the absence of a protein, or flexibly or rigidly in the presence of protein. But in either case, we end up with a set of molecules aligned in a relevant way. What are we going to do with these? Well, one of the earliest breakthroughs, I think, in computational drug design was the 3D QSAR approach pioneered by Dick Kramer, when he was at Tripos. The Colfer approach takes a set of aligned molecules, builds a grid around them, computes properties on the grid, and does some statistics magic to build a model that predicts affinity. We call it machine learning these days. The grid aspect of Confa introduces some pathologies that aren't always desirable. So we thought, well, could we use a simpler way to assemble a data matrix for statistical analysis? And we decided we just look at a much simpler approach, which is the similarity between the molecules in the training set, where we're looking at similarity from rocks using shape and color, from in shape and electrostatics using our engine Eon, or boring old graph similarity. So we compute matrices of similarities, and we pass those into a couple of statistical methods, KPLS, the same statistical method used in Confa, and Gaussian process regression. We'd end up with 2 models and 2 sets of 3D similarities plus a 2D model. We then pass all 5 into a consensus, and that's the prediction from 3D QSAR. So, as an affinity prediction method, this approach based on 3D molecular similarity works very well. Some recently published work from Bernd Kuhn and colleagues revealed this PDE-10A dataset. We took that dataset, applied it to a whole set of literature methods, and we're very pleased to see that our work, far right-hand column, works very well. We get performance equivalent to or superior to all the other techniques applied, including some fairly clever neural networks, a convolutional neural network, and potential network. We're also, second to right column, pleased to see that 3D outperforms 2D here. There's more relevant information in 3D than 2D. But there's more. An important part of the KPLS model building is it produces a set of propensities in space. We've got points in space where the model says, put a shape here, the activity will increase. The model says, put an acceptor here, points in magenta, you'll increase activity. Put a donor here, you'll increase activity, shown in gold. So that is a very useful thing to help spark imagination for chemists to build vector molecules. But we can take it a step further still, and we can essentially boil down, distill all that information into this. This is a set of color features embedded in a shape, which I said 25 minutes ago. That's our core representation of a molecule. This is a representation that can't be matched by any set of atoms joined together by bonds. Features are arranged in ways that can't be accessed all at the same time by a single accessible molecule, but they represent all the features that are contributing positively to the binding affinity that we're trying to model. So as a shape with a set of color features that's sort of got this distilled set of knowledge in it, molecular whiskey, I'm going to call it, we can consume this set of color features embedded in a shape directly in rocks as a virtual screening query. So what we did is did that and compared it to using several different queries from the same protein target, ran each query, 5 queries separately, took the best performing single molecule query and compared it to our distilled piece of molecular whiskey magic. And I'm delighted to be able to say that we actually get better virtual screening performance from the distilled molecular whiskey query than any individual molecule that's active on the same target. So this is great news. We've got a method that now not only produces predictions of affinity across small scale, we can use it for large scale virtual screening at relatively high speed. So I hope to have been able to convince you today that the shape and color representation is powerful across all kinds of different applications. The simple, small molecule similarity and scoring, for protein binding site similarity and scoring, for protein ligand complementarity at large scale with FRET and hybrid, lead optimization complementarity, docking for post-prediction in lead opt, and then the 3D QSAR approach, again, 3D similarities, both to drive machine learning for affinity prediction and to generate our molecular whiskey magic rocks query that works very well in larger scale virtual screening. So as a lifelong Tolkien fan, for me, shape and color is definitely the one ring. It is the one thing that brings a lot of disparate problems together, and we can solve all these different problems under a single. So thank you very much for your attention. And if you have any questions, I'd be happy to take them now.
Thank you, Paul. That was awesome. We have a few questions for you. So I think I'll just start reading them out to what I see here on my screen. It's one, you mentioned X-ray conformations a lot. What happens if I do not have the X-ray conformation?
Quite very good question. Yes. So it turns out, I didn't show the data here. It turns out it actually doesn't make a lot of difference to rocks whether the input conformation for the query comes from experiment, comes from crystallography or cryo-EM, or whether it comes from computation. So We published, actually, you and I published this work a little while ago. We don't actually need the crystal conformation. If we generate, at least with our conformation generator, omega, you take the single lowest energy conformation you can find, compare its virtual screening performance to the crystallographic conformation for the same molecule. The performance of the omega generated conformation is a little bit lower than the X-ray, but the difference is really quite small. So obviously the labor involved in getting a crystal conformation is enormous. The benefit for having that crystal conformation is definitely there on average, but the difference is small enough that for me, it's not worth waiting. If you have an interesting molecule coming out of an assay, just compute the conformation, run the screen, and you're going to get almost as good a results as if you'd waited the months it would take to get the co-crystal conformation. So if you don't have it, don't worry.
Yeah. Another one. I ran many virtual screens over my career, and hit rates were always very low. Why do virtual screens have such low hit rates?
Oh, goodness. Okay. Well, I think we could think of several things. I did make a big deal about the size of chemical space, but none of the chemical spaces that people generally screen against are designed with reference to any particular protein type or any particular individual protein. So the database is full up of molecules that aren't really relevant to the problem that you're trying to solve. One. The same way the scoring functions that any virtual screening tool uses, ligand-based, structure-based, 2D, 3D, the scoring they use is almost by definition not relevant to your problem. It's a generic scoring function. In ligand-based, it's usually molecular similarity. It's very general, but it's generic. It doesn't necessarily focus on your problem. And thirdly, and unfortunately, the false positive rate is a problem, that you've got so many more inactive molecules in the database that even if your scoring function is very, very good at discriminating between actives and decoys, actives and inactives, there is still a rate at which it incorrectly assigns an inactive to act. And then you can very easily overwhelm the actual actives in your database with all the false positives because of the large number of inactives that you've got in the database instead. So we're really sort of up against it. And to some degree, it's amazing that virtual screens work at all, given all the approximations we make to the scoring and the necessary but annoying false positive problem. So there's three, at least, things that are working against us there.
Right. I have one more here. Back to crystal structure somehow. Are the A-fit and shape-fit approaches of fitting related?
Oh, yes. Sorry, yes, I hope I've made that clear. Yes, so A-fit is fitting into density, and it's fitting a molecule flexibly into density. Density obviously has no color. It's just a shape of density in space. So we just fit shape to shape in A-fit. For the shape-fit engine, we're actually taking usually a crystallographic ligand, though you can put it there computationally, and we're flexibly fitting a new molecule in based on matching shape and color, because there's shape and color in the molecule. So absolutely related in the approach. A-fit can only use shape because there's only density. Shape-fit uses both shape and color because we're fitting to an actual molecule.
But in the end, the strain goes in as well, just to not fit something too strained into the original shape.
We tried very hard not to produce steeple posts.
Excellent. I think with that, also due to time, thank you very much, Paul, for today.
Thank you.
Any more questions, any other questions we have here will be answered by email. So thank you very much for sending them in. And with that, I hope to see everybody in our next presentation, in the next webinar, and see you then. Bye-bye.